A longitudinal study published in Nature Aging in 2022 tracked mitochondrial function across 847 individuals from age 25 to age 65. The researchers measured ATP production capacity, membrane potential stability, and electron transport chain efficiency at 5-year intervals. The inflection point appeared at age 31, not approximate, specific. Before 31, mitochondrial decline averaged 0.8% per year. After 31, that rate jumped to 2.4% annually through age 45, then accelerated further. The pattern held across all tissue types measured. The mechanism behind that acceleration is what we need to understand. The standard model teaches that aging is gradual and linear. Cellular decline accumulates slowly over decades. Mitochondrial function follows the same gentle slope. And that model dominates medical education and shapes how we think about biological aging. It is not incorrect. It is incomplete. The 2022 Nature Aging data documents something different. A threshold effect, a gear shift in the rate of decline. The transition happens in the early 30s, and the biological consequences compound from that point forward. Why does this matter? Because mitochondria are not just energy factories. That metaphor undersells their role. Mitochondria regulate apoptosis, calcium signaling, redox balance, and inflammatory response. When mitochondrial function declines by 40% between age 31 and age 50, as the longitudinal data shows, that decline affects every system downstream. Cognitive performance, muscle recovery, immune resilience, metabolic flexibility. All of those capacities are mitochondrially gated. Yet the threshold at 31 is not arbitrary. It marks the point where cellular energy production begins to limit systemic function. The research documenting this pattern comes from multiple independent labs. A 2020 study from Johns Hopkins measured mitochondrial DNA copy number in 1,200 participants. The decline became statistically significant at age 32. A 2021 paper in Cell Metabolism used phosphorus-31 magnetic resonance spectroscopy to measure ATP synthesis rates in skeletal muscle. The rate dropped 18% between age 30 and age 40 in healthy, physically active adults. A 2023 study from the University of Copenhagen tracked cardiorespiratory fitness and mitochondrial density in endurance athletes. Even in this highly trained population, mitochondrial capacity peaked at age 29 and declined measurably by age 33. Now, the pattern is reproducible across methodologies and populations. What drives the threshold at 31? The mechanism centers on mitochondrial quality control. Mitochondria replicate through fission. Damaged mitochondria are cleared through mitophagy. That balance between replication and clearance maintains the functional pool. The regulatory proteins governing this process are NAD+ dependent. NAD+ is a coenzyme required for mitochondrial biogenesis and the activation of sirtuins, which coordinate mitochondrial repair. NAD+ levels peak in the mid-20s. A meta-analysis published in Aging Cell in 2021 reviewed 31 studies measuring NAD+ across age groups. The data showed a consistent 15% decline between age 25 and age 35, with the steepest drop occurring between 30 and 34. That decline in NAD+ availability reduces the activation of SIRT1 and SIRT3, the sirtuins responsible for mitochondrial maintenance. When those pathways slow, damaged mitochondria accumulate. The functional pool contracts. ATP output declines. That is the biological architecture behind the threshold. The electron transport chain operates through 5 protein complexes embedded in the inner mitochondrial membrane. Electrons move from complex 1 through complex 4, pumping protons across the membrane to create a gradient. That gradient drives ATP synthase. The efficiency of this process depends on membrane integrity. As mitochondria age, reactive oxygen species damage the membrane lipids and protein complexes. The damage accumulates faster than the repair mechanisms can compensate once NAD+ levels drop below a critical threshold. A 2022 study in free radical biology and medicine measured complex efficiency in muscle biopsies from 340 participants. Efficiency remained stable through age 30, then declined 3.2% per year through age 45. The inflection point aligned precisely with the NAD+ decline documented in earlier studies. Everything points to the same mechanism. Mitochondrial decline is not evenly distributed across tissues. The brain and heart are disproportionately affected because those organs have the highest energy demands. A neuron requires approximately 4.7 billion ATP molecules per second to maintain ion gradients and support synaptic transmission. When mitochondrial output declines by 20%, neuronal function becomes energetically constrained. A 2020 study from Stanford used functional MRI to measure brain metabolism in adults aged 25 to 55. Prefrontal cortex ATP synthesis rates declined 22% between age 30 and age 45. That decline correlated directly with performance on executive function tests. Processing speed, working memory, and cognitive flexibility all showed measurable declines that tracked mitochondrial capacity. The mechanism is not mysterious. Neurons running on reduced ATP operate less efficiently. The subjective experience of that inefficiency is what people describe as brain fog or mental fatigue. And skeletal muscle follows a similar pattern, but with a slightly later threshold. Muscle mitochondria are more resilient because muscle tissue maintains higher baseline mitophagy rates. A 2021 study in the Journal of Applied Physiology measured mitochondrial density in the vastus lateralis of 520 adults. Density remained stable through age 34, then declined 1.8% per year through age 50. The decline accelerated to 3.1% per year after age 50. The functional consequence is reduced exercise capacity. VO2 max, the maximum rate of oxygen consumption during exercise, is mitochondrially limited. A meta-analysis of 94 studies published in Sports Medicine in 2022 found that VO2 max declines approximately 10% per decade after age 30 in sedentary individuals. In trained athletes, the decline is smaller but still present. The mechanism is identical. Fewer functional mitochondria means less capacity to generate ATP during high-intensity effort. The immune system is the third system significantly affected by mitochondrial decline. Immune cells, particularly T cells and macrophages, require substantial ATP to mount an effective response. A 2023 paper in Immunity documented that mitochondrial dysfunction in T cells begins in the mid-30s and correlates with increased infection susceptibility and reduced vaccine response. The study measured mitochondrial membrane potential in T cells from 680 participants. Membrane potential declined 12% between age 30 and age 40. That decline reduced the capacity of T cells to proliferate in response to antigen challenge. But the clinical manifestation is longer recovery times from infections and reduced adaptive immunity. The biological mechanism is energy limitation at the cellular level. Research has identified a natural compound that supports this neural and mitochondrial mechanism at the cellular level. It is not a drug. It is not a treatment. It is a natural formula built from published research on brain and cellular energy metabolism. If you want to understand how it works, the link is in the description. This is a sponsored recommendation and individual results vary. The research suggests that mitochondrial decline after age 31 is not an on-off switch, but a progressive constraint. The body does not fail suddenly. It becomes less energetically efficient over time. That inefficiency manifests differently depending on which system is most heavily loaded. For someone whose work is cognitively demanding, the first noticeable effect may be reduced mental stamina. For someone who trains intensely, it may be slower recovery or reduced power output. For someone managing chronic stress, it may be increased susceptibility to illness or longer recovery from infections. These are not separate problems. They are different expressions of the same underlying constraint. Mitochondrial capacity is the bottleneck. What the mainstream medical model treats as separate age-related conditions are often downstream consequences of mitochondrial insufficiency. Sarcopenia, the loss of muscle mass with aging, is mitochondrially mediated. Cognitive decline in the absence of neurodegeneration is mitochondrially mediated. Increased inflammation and reduced immune function are mitochondrially mediated. The standard of care addresses these as independent conditions because they present independently. But the biological architecture connects them. The constraint is energetic. Everything else follows from that constraint. The distinction between disease and optimization is critical here. Standard laboratory ranges are designed to identify pathology. A fasting glucose of 105 milligrams per deciliter is prediabetic. A glucose of 95 is normal, but normal is not optimal. Mitochondrial function that supports baseline health is not the same as mitochondrial function that supports peak performance. The research we are discussing operates in that gap. The threshold at age 31 does not mark the beginning of disease. It marks the beginning of a progressive decline in energetic capacity that eventually manifests as the functional limitations people associate with aging. Those limitations are real, they are measurable, and they are mechanistically tied to mitochondrial output. The viewer watching this is likely somewhere in that 31 to 50 window. The data suggests you're operating on reduced mitochondrial capacity relative to your mid-20s baseline. That reduction is not catastrophic, it is incremental. But incremental reductions compound. A 2% annual decline sounds manageable. Over 15 years, that is a 30% reduction in ATP synthesis capacity. That reduction changes what your body can sustain. The mechanism is precise. The timeline is documented. The intervention window is now. If this gave you a new way to think about your biology, leave a comment below. I read every one. And if you found this useful, the single best thing you can do is follow the channel. We decode a new mechanism every day. Everything you need is in the description and in the first comment. Before you go, we built a free 2-minute biological wellness assessment that reviews 4 body systems and gives you a personal pattern score. The link is in the bio, in the first comment, and in the description. 2 minutes. It puts numbers to everything we just discussed. The next mechanism we decode is how your brain compensates for that mitochondrial decline and what happens when that compensation fails. The 2022 Nature Aging study included a secondary analysis that most people missed. The researchers measured not just mitochondrial output, but mitochondrial network morphology using electron microscopy. Mitochondria exist as dynamic networks, not isolated organelles. They undergo continuous fusion and fission to adapt to energy demands. In participants under age 30, mitochondrial networks were highly interconnected, with an average of 18 fusion events per cell per hour. After age 35, fusion frequency dropped to 11 events per hour. By age 45, it was down to 7 events per hour. This matters because fused mitochondrial networks are more efficient at ATP production and more resistant to oxidative damage. Fragmented mitochondria produce less ATP and generate more reactive oxygen species per unit of substrate oxidized. The proteins regulating mitochondrial fusion are called mitofusins. MFN1 and MFN2 are the primary regulators. A 2023 study in Cell Reports measured mitofusin expression in skeletal muscle biopsies from 410 adults across age groups. MFN2 expression declined 28% between age 30 and age 50. That decline directly correlated with increased mitochondrial fragmentation and reduced oxidative capacity. The mechanism is straightforward. Without adequate mitofusin activity, mitochondria remain fragmented. Fragmented mitochondria are less efficient and more prone to damage. The damage accelerates the decline in functional capacity. This is a feed-forward loop. Reduced NAD+ leads to reduced mitofusin expression. Reduced mitofusin expression leads to mitochondrial fragmentation. Fragmentation increases oxidative stress. Oxidative stress further reduces NAD+ availability. The cycle reinforces itself. The liver is particularly vulnerable to this cycle because hepatocytes have among the highest mitochondrial densities of any cell type. A single hepatocyte can contain over 2,000 mitochondria. The liver is responsible for metabolic regulation, detoxification, and lipid processing. All of those functions are ATP intensive. A 2021 study in hepatology measured mitochondrial function in liver biopsies from 290 adults without liver disease. Mitochondrial ATP production capacity declined 19% between age 32 and age 48. That decline correlated with increased liver fat accumulation and reduced insulin sensitivity. The mechanism is energetic. When hepatocytes lack sufficient ATP to process incoming lipids, those lipids accumulate as intrahepatic fat. Non-alcoholic fatty liver disease is partially a mitochondrial insufficiency disorder. The standard model treats it as a lipid storage problem. The mitochondrial model reveals it as an energy production problem. Emerging research from 2023 and 2024 has begun exploring the connection between mitochondrial decline and circadian rhythm dysregulation. Mitochondria contain their own circadian clocks independent of the suprachiasmatic nucleus. A study published in Science Advances in 2024 found that mitochondrial circadian amplitude declined significantly after age 35. The researchers measured the expression of clock genes within mitochondria across 24-hour cycles. In participants under 35, mitochondrial clock gene expression showed robust oscillation with a peak to trough amplitude of 4.2-fold. In participants age 40 to 50, that amplitude was reduced to 2.1-fold. The functional consequence is reduced temporal coordination of mitochondrial ATP production with cellular energy demands. When cells become less efficient at matching energy supply to energy demand throughout the day, that mismatch manifests as fatigue that does not align with activity levels. The subjective experience is energy that feels unpredictable or unreliable. The blood-brain barrier adds another layer of complexity. Endothelial cells lining the brain capillaries are highly mitochondrial because maintaining the barrier requires continuous ATP input. A 2023 study in Nature Neuroscience measured blood-brain barrier integrity and mitochondrial function in brain endothelial cells. Mitochondrial output in these cells declined 16% between age 30 and age 45. That decline correlated with increased blood-brain barrier permeability. When the barrier becomes more permeable, peripheral inflammatory molecules gain access to the central nervous system. Neuroinflammation increases. Cognitive function declines. This is not speculation. The mechanism has been directly measured. Mitochondrial insufficiency in brain endothelial cells compromises barrier integrity. Barrier compromise allows inflammatory signaling. Inflammation impairs neuronal function. The chain is complete. What makes this research particularly relevant now is that the interventions being explored in current trials target these mechanisms directly. A 2024 phase 2 clinical trial published in Cell Metabolism tested a combination of NAD plus precursors and mitochondrial membrane stabilizers in 180 participants aged 35 to 55. The intervention increased mitochondrial ATP production capacity by 14% over 12 weeks. Participants also showed improvements in VO2 max, cognitive processing speed, and inflammatory markers. The trial was not designed to treat disease. It was designed to test whether mitochondrial function could be enhanced in healthy adults experiencing age-related decline. The results suggest it can. The mechanism is supportable. The timeline matters. Waiting until mitochondrial function has declined 50% makes restoration harder. Intervening at the threshold, when decline is measurable but not yet severe, offers a different opportunity. That threshold is age 31. The research is precise about the timeline.